Thank you, Nazil, for the invitation. It's an honor to be present here in front of the stalwarts of hepatology to present the work. So when we submitted the manuscript to Nazil, we had the complete data on metabolomics. But now we have completed the work both with metabolomics and RNA sequencing and ready to submit to the journal. So I stand on behalf of the entire team, and the work is ready to be submitted to a peer-reviewed journal now. So I realize that I don't need to give a background information of how important it is to understand the pathophysiology of MASH and identifying a novel molecular target for the therapeutic strategy. So let me pitch to the rationale for the study. So this started when I took up a postdoctoral position with Dr. Arun Sanyal at VCU in the United States, where we saw that there was an upregulation of a transcription factor called AATF, apoptosis antagonizing transcription factor, which we observed that it was upregulated in all the Western diet sugar water fed mice when compared to the control. So that was the project taken up for my postdoctoral work. We identified what was the reason for this upregulation in the MASH. We were the first to identify the role of AATF in MASH, found how is AATF upregulated in MASH, and then did a xenograph study using control AATF versus the knockdown AATF, elucidated the mechanism with the xenograph model, and it got published in Hepatology. And then the decision was taken, like I wanted to move back to India with the prestigious Ramalingaswamy Re-Entry Fellowship offered by the Department of Biotechnology. So this is one of the projects which I'm continuing as a faculty here at JSS Medical College, very close to heart, because many questions remain unanswered to what was the mechanistic role of AATF in the pathophysiology of MASH. So we have seen the upregulation of AATF in the cellular model of MASH, moving on with the preclinical model. We have also seen upregulation of AATF in the human samples of MAFL and MASH. So knowing that, the first question was, what happens if we inhibit or silence the AATF in the preclinical model? For this, we used a C57 Black 6 mice from Charles liver, and then the diet was procured from the Research Diet US, and we synthesized or have a vector which was AAV8 vector, where AATF was silenced with the TBG promoter, making it very specific to the liver. So this was a preclinical study carried out for 24 weeks, and obviously we have the phenotypic notification, biochemical analysis, histology. The crux of the study would be the RNA sequencing and the metabolomics. So the first thing to make sure that the experimental work is to check the expression of AATF. Yes, as you see, there was inhibition of AATF expression in the liver, but not in any other organ, and the protein expression is also being done. So moving on, I'll be presenting only the key data, given that only 9 minutes is allotted for the talk. So the phenotypic data, biochemical data, I'm going to skip over very fast because the crux of the study is metabolomics and RNA-seq. So as you see, there was the impact of silencing hepatic AATF, which ameliorated the Western diet sugar water-induced obesity, where you see in the reduction of body weight, liver weight, and adipose tissue weight, and insulin resistance. Following, we do a series of biochemical analysis and histology, and all of which also showed that silencing of hepatic AATF prevented liver injury and improved the steatohepatitis. The crux of the study was understanding how the RNA sequencing and metabolomics resulted. So this data is very critical in identifying that there is no variation in the sample, be it inter-sample or intra-sample. The data was very clean, where you can see the distinction among the groups, and also the sample-sample variation is nil. So this is where we compared the RNA sequencing data versus the metabolomics data. In the RNA-seq data, so throughout the study, we just didn't depend on the RNA sequencing. We validated certain targets, both by the PCR expression as well as Western blot and ELISA wherever required. So as you see, the cellular stress, inflammatory response, and the fibrosis was alleviated when we silenced hepatic AATF. Moving on, we also saw that we had a question whether there would be an effect of silencing hepatic AATF in lipid metabolism. Has hypothesized, yes, when we silenced AATF, there was reduction in the lipid metabolism, in fact, the lipid synthesis. So then we started analyzing and correlating the RNA-seq data with the metabolomics data. So the metabolomics was an untargeted metabolomics that we performed. So when we took the lipid class, the superclass, as well as the subclass, we saw that glycerophospholipid metabolism was upregulated in the silenced AATF. And also the lipid subclass was also very clear. So is it also correlating with the RNA-seq data? Yes. The silencing of AATF upregulated the glycerophospholipid metabolism in preclinical study. We also validated certain glycerophospholipid metabolites using the metabolomics data. The interesting data here is, there is a software called MetaboAnalyst where we can input all the clinical variables and see whether there is a positive or a negative correlation and how is it related with the glycerophospholipid metabolism. The data was very evident and positive, showing that silencing of hepatic AATF upregulated the glycerophospholipid metabolic pathway. So this is a parameter where we correlate the clinical variables and also relating it to the glycerophospholipid metabolism. Moving forward, we asked another question. So is it only limited to glycerophospholipid metabolism or other metabolism are also associated? Favoring the hypothesis, we saw there was upregulation in the mitochondrial beta oxidation and alpha-linoleic acid metabolism. So these are the metabolomics data. So was it also happening in the RNA-seq data? Yes. We saw that the fatty acid beta oxidation was upregulated in the silenced preclinical model versus the control. So with this, we could identify very clearly that silencing hepatic AATF could also promote fatty acid metabolism, fatty acid beta oxidation. So to summarize, this was a preclinical study where silencing of AATF was carried out for 24 weeks, where we saw an upregulation in the Western diet sugar water fed mice, while silencing AATF ameliorated the liver injury as well as the other phenotypic data, and along with reducing cellular stress, inflammation, and fibrosis. With metabolomics and transcriptomic data integrated, we found that the glycerophospholipid metabolism, alpha-linoleic acid metabolism, fatty acid oxidation, which are very critical in reducing the fatty liver, was upregulated in silencing AATF versus the control. So with due respect to time of 9 minutes, I have only presented the key data. So I'll be happy to answer any question at the end of the session. So an interesting study that we wanted to see was, so these are two databases that we put forward, that is the BioGrid database as well as string database, and combine both of them. So this basically operates on integrating the already available database in the system. So as we saw, there was a lot of interacting factors of AATF with ribosome biogenesis and DNA replication. And for the first time, we were able to identify that there's an indirect association of AATF with CD74, which is known to play a key role in reducing inflammation in MASH. So this study was led to the identification of how AATF could interact with an existing transcription factor CD74, which is involved in the fatty acid metabolism. Of course, there are many other questions that remain unanswered. I acknowledge the team and also my mentors. And of course, thank you all for your kind attention. I'm right on time. Happy to take up any questions.
Excellent work. Two
Thank
questions. One is on inflammatory markers and the inflammatory histology, what it showed. And similarly for fibrosis, what were the, I mean, it was not clear from your presentation whether it had any effect.
Yes.
And what was the effect?
Yes.
Both histologically and other markers.
Yeah, I'll just go through in detail. Because of the time, 9 minutes of allotation, I skipped that portion. Can I have it on displayed on the slide, please? So histology, it was blindfolded and this was been analyzed by the pathology as JSS Hospital. We saw that the steatohepatitis was ameliorated. As you see by the scoring which was done by the pathologist, we see a reduction in the MASLD. And this was also consistent with the fibrosis data that we have seen. both with RNA-seq data and also the biomarkers that was examined in the laboratory.
Rakesh?
Yes, sir. For preclinical study? Yes, it was a chow diet versus the Western diet. So the comparison we did was how it... Yes, sir, of course. I think I focused mainly on the Western diet and the preclinical model where we saw, it is a basic understanding here, where we use the TBG blank versus the ATF siRNA. Sorry, I think because of the 9-minute slot, I would not go in detail for each and every experimental detail.
2 more questions. Divya first. Nice work.
Thank you, sir.
What happened to the mono and diacyl glycerols in the liver? Did you test? If you're not making triglyceride, where is the pathway blocked?
Okay, so as I said, this was an untargeted metabolomics. We get a complete profile of all the triglycerides when we do...
If you're not making TG, what are you making? Mono or di? 8, 10, 12 amino acids?
So this question remains unanswered to us because...
It is not
yes,
addressed. So
yeah,
that is fundamentally, you're not reaching to tri, so it must be di or something.
Di or mono, yes.
The second is you're showing everywhere soluble AATF. So what is the soluble vis-a-vis the tissue AATF?
Pardon,
you are
sir?
showing the SL, soluble.
SI. It's an siRNA.
Oh, it is siRNA.
Yeah, siRNA very specific to the liver.
The third part was what is the efficacy of inhibition?
As you see in the Western blot data, it was 80% of inhibition.
Inhibition.
Yes.
So what is the normal function of AATF? If it is so bad?
AATF is known to be playing a role in DNA replication and ribosome as such. So we have seen it upregulated in certain cancers, and we were the first to show in hepatocellular carcinoma as well. So we don't know what is it doing at Massilley.
I thought it was a normal, good thing, but we have more questions. We can discuss later. Please.
Thank you for that presentation, Dr. Divya. I have 2 questions.
Yes.
First is that your study group size of n is equal to 3. Do you think it's adequate to make these conclusions? And second question is, there are certain kind of basic differences between the human liver and mouse liver. And have you compared your findings with any of the RNA-seq or lipidomics or metabolomics data sets that exist from human studies?
Okay, to answer your first question, the model which is used here is been tested already by Dr. Scott Friedman, where they have done an RNA sequencing, and it has clearly shown that the histology of the mice is very much equivalent to the human. Regarding with N sample, with the preclinical study, we have used N is equal to 15, but when you take it forward to RNA sequencing and metabolomics, because of the cost issue, we stick on to N is equal to 3 or 4. Otherwise, with all the phenotypic data, biochemical analysis, and molecular biology data, it's N is equal to 15 per group.
Okay. Thank you.
Yes.